Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. First of all, I would like to introduce myself. My name is Hertien ABS70. I work together with Rian Kuniawan Sahinsia. Today, I would like to give a presentation of my research with the title Plankton Biodiversity and Freshwater Ecosystem Feasibility as a Learning Resource Situ Bagendit. In this presentation, I would like to tell about introduction, research method, result and discussion, and the last is the conclusion. The best way to learn science optimally, first by utilizing local resources in the environment, second making direct observation of the surrounding environment, the third creating a good and active learning environment, and the fourth, innovation and curiosity as the basis of science learning. Environmentally friendly learning is think globally and act locally, which means learning environmentally oriented local to achieve global thinking skills, and it can be done by solving problems in the local environment. The statement will be implemented in the course of Freshwater Biology in the Biology Study Program, the fifth semester students, because Freshwater Biology is an interdisciplinary science course that involves biological, chemical, social, and environmental sciences. Learning in class in theory is not enough. It must be supported by multidisciplinary field lectures by utilizing learning resources. There are 354 lakes or situ in West Java province which can be used as a learning resource. Situ Bagendit has an area of between 10 to 50 hectares with natural boundaries and very clear administrative boundaries. Situ Bagendit is located at an altitude of 800 meters above sea level and has a general configuration of flat and hilly land. The existence of Situ Bagendit is a nature tourism. It has been widely used for various activities. Therefore, the quality of lake water will experience changes that will affect the diversity of plankton. This is the location address of Situ Bagendit, Jalan KH Hasan Arif, Banyuresmi. Situ Bagendit tourist attraction is in the Bagendit area, Banyuresmi district, Garut Regency. The lake is located in the middle of a cool and peaceful village with an administrative boundary. To the north bordering Banyuresmi village, south with Cipicung village, east with Binakarya village, and west with Sukamukti village. For this research method, in this research, 4 stages were carried out, namely A. Study of the biology plankton composition in Situ Bagendit, B. Chemical analysis of Situ Bagendit water, C. Preparation of research-based material, D. Compilation of the plankton identification book, and E. Situ Bagendit feasibility study as a learning resource. This map location show you the distance between Bandung and Garut. So it takes 2 or 3 hours by car from Bandung to my study site in Garut. For the sampling location, there is 6 points. The first point, this is a halfway between the sand mine and the tourist attraction. Point 2, this is the floating stalls. Point 3, the final part where Nelumbo nucifera is overgrown. Point 4, the initial part of the place where Nelumbo nucifera is overgrown. Point 5, Bunbun fishing. Point 6, Bunbun fishing and the farthest point from tourist attraction, or it is the inlet area. This picture show you about the Situ Bagendit. Figure 1 show you the Bunbun fishing. So there are two different Bunbun fishing. They use from bamboo to collect the fish. And then for figure 2, this is a floating stall that we can pass to this place to buy some drink or some snack. And there is a book of learning media about plankton that I write down for helping the student study in Situ Bagendit. In this book, there is 5 subchapter. There is a biology and ecology of plankton, the biodiversity of plankton, the adaptation of plankton, the bioindicator of plankton, and the play role of plankton in freshwater ecosystem. This is the physical and chemical analysis water. We have 6 parameters for physics and chemical analysis of water. There is a temperature, light, pH water, dissolved oxygen, carbon dioxide, and biological oxygen demand. And this is the equipment that we have to bring to the field to measure the water analysis. And this is the biological analysis of plankton. The first thing we have to do the lake water sampling, and then preservation of lake water samples, plankton identification and calculation, and the last is plankton data analysis includes diversity, dominance, and abundance using Shannon-Weiner in ODAM 1993. And this is the guidebook of plankton. So the student, if they go to the field in the Situ Bagendit, they can use this good guidebook of plankton for identification. And this table show you the species of phytoplankton and zooplankton in 6 site location. And this is the plankton data result. Plankton abundance ranges types of phytoplankton 6 classes with 26 types, and zooplankton as many as 8 classes with 19 types. The distribution of plankton types is not evenly distributed at 6 sampling points. The most abundant plankton found in location is Microcystis smithii comarec with numbers 11,867, followed by Crucigenia species 2,133, Eglena species 1,733, Scnedesmus acuminatus 1,333, and Crucigenia lauterborni 1,333. This is the result of water chemical analysis data. We have 9 parameters: ammonia total, phosphate total, chemical oxygen demand, nitrite, nitrogen organic, chlorophyll A, the average of light, and the average of total N. And this is the method that I use based on the APHA standard international. And we measure the water only in the 3 location site because of the limited budget for doing the analysis. And this is the table of lake tropical status. This table show you how to identify, how to determine the status trophic. So we have 4 status trophic: oligotroph, mesotroph, eutroph, and heterotroph. And then there are some parameter: average total N, average total phosphate, average chlorophyll A, and average light. So if we have this data, so we can determine what kind of status trophic of the lake. For example, if the average total N less than 650 microgram per liter, so it is considered as a oligotrophic. But if the average total N higher than 1,900, it means the lake is considered as hypertroph. This lake in eutrophication status. In visual, we can see that there is a lot of floating plant around the Situ Bagendit. So it can be indicated that the lake is in the eutrophic status. And we study also about the feasibility of Situ Bagendit. So we have 6 criteria: easy of access, safety, cost, time, learning media, and the aspect of science in studying plankton in Situ Bagendit. For the access, here from UPI campus to Situ Bagendit, it can be traveled by motorized vehicle or public transportation. So it means that it's very easy to get there from campus to Situ Bagendit. And then Situ Bagendit is safe from wild animals, and it is a tourist location. The cost to get the Situ Bagendit is affordable, so it means very cheap. And the entrance ticket is about 25,000 rupiah. And also we can go to the Situ Bagendit using boat or raft, and we have to pay 100,000 per day, accommodating 20 people. And the fee takes 2 hours from UPI to Bandung. Media is available in the form of plankton identification, guidelines, and teaching materials about the Situ Bagendit and plankton. So this book or this media will help the student to study about Situ Bagendit. And then another aspect of science in studying plankton, that we found 6 types of phytoplankton species with 26 types, and zooplankton as many as 8 classes with 19 types. So the total number of individuals plankton is 3,512. And these some plankton are bioindicator of eutrophication status. So it means if we want to learn about the diversity of phytoplankton, so Situ Bagendit is available to study there. For the conclusion, water quality of Situ Bagendit is hypertroph or high organic pollution. It is indicated with high organic pollutants and abundance of plankton as a bioindicator. It consists of 25 types of phytoplankton and 80 types of zooplankton. This plankton biodiversity in Situ Bagendit is an alternative that Situ Bagendit may be used as a learning resource, while the level of eligibility of the Situ Bagendit, which includes ease of access, security, cost, time, teaching media, aspect of science, and the benefits of learning resources, may support the learning process. For the acknowledgement, I would like to thank to the funds available at the University of Indonesian Education Fiscal Year 2018. And also I would like to thank to Rian Kurniawan, Muhammad Naufal, Raka Sabar, and Sonia Putri, who helped in data collection in the field. Thank you very much for your attention.